2A. Benedict's solution in A remains blue, no color change, negative result, as there is no maltose breakdown of starch. Benedict's solution in B turns red, changes color, positive result, in the presence of maltose. Amylase breaks down starch into maltose. Maltose is a small, soluble molecule. Maltose is able to pass through the dialysis tubing by diffusion from an area of high concentration to low concentration down a concentration gradient. 2B. Pepsin protein amino acids peptides. Lipase fats, oils, lipids, fatty acid and glycerol. Trypsin protein amino acids peptides. Maltase, maltose, glucose. 2C. Villi, microvilli. 2I. Any 2 from bile, hydrochloric acid, mucus, water, saliva. 2AII. Name of enzyme. Organ where enzyme is secreted. Organ where the enzyme acts. Products of digestion. Involving this enzyme. Amylase, salivary glands, mouth buccal, cavity, simpler, sugars, maltose, glucose, pepsin, stomach, gastric glands, stomach, amino acids, lipase, pancreas, small intestine, duodenum, ileum, fatty acids and glycerol, maltase, small intestine, epithelial lining of the small intestine, glucose. 2B. Any 6 from 39 plus or minus 2 is optimum temperature. Highest activity is 2500 U per milligram protein. Steeper decrease in activity after optimum. No activity after 62 degrees Celsius plus or minus 2. Increased temperature causes increased kinetic energy. Increased frequency of effective collisions. Forming enzyme substrate complexes. Denaturation occurs at any temperatures above optimum temperature. Active site changes shape. No longer fit into substrate, complementary to substrate. Idea that both enzyme and substrate are proteins and may change. 6. Each unit must be stated at least once. Don't accept on wrong or no units. 2C. Any 2 from lactose is found in milk. Lactase breaks down lactose. Young babies are dependent on lactose milk for their nutrition. 2CII. Loss of watery feces. 2CE. Any 3 from cholera bacterium release toxins. Causes chloride ions to be secreted from the body. Chloride ions in small intestine, duodenum, ileum. Ref to osmotic movement lowers water potential in gut. Water moves into gut or more water remains in intestines, gut. 1 by any 4 from pH decreases, solution becomes acidic. pH changes because fatty acids are produced. Lipase digests, breaks down fat. Fatty acids produced faster in test tube C than B. Bile emulsifies fats, converts large particles of fat to small particles. Bile increases the surface area for lipase action. 1BII. Compare with tubes B and C to assess effect of lipase and or bile. Shows that bile does not chemically digest fats, does not make solution acidic. Shows that lipase enzyme is required for breakdown of fats in milk. Control experiment. 1C line drawn showing that decrease after optimum is steeper than increase. MP1 line does not have to start at the origin or end at the x-axis. Label line to point where line meets the x-axis and label indicating that all the lipase is denatured. MP2 line must meet the x-axis. 1D. Any 3 from ref to specificity. Only substrate for lipase is fat molecules. Shape of active site is not complementary to protein, complementary to fat. Protein cannot fit into bind to active site lipase enzyme. Enzyme substrate complexes cannot be formed. Soluble stomach, hydrochloric acid, named microorganisms, pathogens, pancreas, small intestine, alkaline, bile, bile salts, emulsification. 6A. Name of enzyme, function, site of action. Pepsin breaks down protein to amino acids, polypeptides, stomach. Trypsin breaks down protein, polypeptides, to amino acids. Peptides, small intestine. Maltase breaks down maltose to glucose. Intestinal, epithelium, small intestine. 6 by a gene, allele. C ribosome. 6 BII, mRNA, a copy of the gene array. Moves out of the nucleus to the cytoplasm. 6b, the sequence of bases in mRNA DNA gene. 6c, ref. To specificity, shape of active site and substrate must be complementary. To enable enzyme and substrate to bind, enzyme substrate complexes to be formed. 0610_S22_Ms_41. 1I. Fungus, fungi. 1AII. Aerobic respiration. 1B. Any 2 from ref. To specificity of enzyme. Shape of active site is complementary to substrate, sucrose. 4. Substrate, sucrose, to bind, fit, enzyme, sucrase, active site. 1c. Total of 6 from max. 4 for description. Sucrase enzyme is active between pH 2 and pH 12. Activity increases and decreases, reaches a peak. Peak maximum or 100% activity at pH 6. Steeper increase between pH 5 to 6, steeper decrease in activity between pH 9 to 10. Minimum 10% activity at pH 12. More activity in acidic conditions, low pH, than alkaline conditions, high pH. Explanation. Change in pH affects the shape of sucrase active site enzyme. At pH 6, most enzyme substrate complexes form. At low, high extremes of pH, enzyme is partially denatured. Ref to substrate molecules can no longer bind with enzyme at low, high extreme pH, so activity decreases. 0610_S21_Ms_43. 1I. Water. 1AII. Nutrient elements enzyme. Products of digestion, products absorbed into blood. Protein. C, H, O, N, SN. Protease, pepsin, trypsin. Amino acids. Fat C, H, O, lipase, fatty acids and glycerol. Lactose. Milk. Sugar. C, H, O, lactase, galactose and glucose, simple sugars. 4 elements for protein and fat for 1 mark. Enzyme and products for protein for 1 mark. Enzyme and products for fat for 1 mark. Enzyme for lactose for 1 mark. 1 by. Any 3 from. Needed for growth, development, strengthening of bones. Needed for growth, development, strengthening of teeth. Prevents rickets. 1 BII. Any 2 from. Required to make hemoglobin, red blood cells. For transport of oxygen. Prevents iron deficiency anemia. 1C. Disease is caused by a pathogen. Pathogen is passed from one host to another. 1CII. Any 4 from. Rubella is caused by a virus. Virus can cross the placenta. Virus can affect embryo fetus. Can cause serious disease, damage, death to baby offspring. Vaccination stimulates long-lasting active immunity in the mother. Immunity gained before they become pregnant. 1CE. Not enough protein in the diet. Adequate energy in the diet. Inadequate diet after breastfeeding finishes. 0610_S20_Ms_41. 2A. 1 mark per row. Substance enzyme products. Starch, amylase, maltose, glucose, simple sugars. Fat, lipase, fatty acids and glycerol. Protein, protease, pepsin, trypsin, amino acids. 2B. Any 4 from. Biological washing powder is more effective at lower temperatures between 10 degrees Celsius and 40 degrees Celsius, 10 degrees Celsius and 43 degrees Celsius. Comparative data quote for the difference at a stated temperature. Biological washing powder removes all stain between 30 degrees Celsius and 40 degrees Celsius. Non-biological removes all stain only at 60 degrees Celsius. Effectiveness is similar at high temperatures between 50 and 60 degrees Celsius. Same trend, below 30 degrees Celsius at low temperatures from 50 degrees Celsius. Idea of effectiveness of biological washing powder decreases between 40 degrees Celsius and 44 degrees Celsius, no such decrease for non-biological washing powder. 2C. Any 2 from. Active site changes shape. Substrate no longer fits into enzyme active site. No enzyme substrate complex, no successful collisions. 2D. Any 2 from. Individual people have different, unique DNA. DNA has genes or alleles have sequences of bases.